So in Alzheimer's disease, we all know there is a senile plaque. Senile plaques is nothing but it is the aggregate of A beta protein present in the outside the neurons. Extraneuronally, there is accumulation of A beta protein, and we all know A beta protein is a derivative of APP. APP stands for amyloid precursor protein. So how this amyloid precursor protein forms A beta subunit will form in we'll see in this today's video now. This is a full-length APP, amyloid precursor protein, and this is a transmembrane protein. So if you take this as a cell, through and through the cell membrane, this protein is arranged. So it is a single-pass transmembrane protein. So this amyloid precursor protein can be cleaved by either by a beta secretase, alpha secretase, or gamma secretase enzyme. The order in which it cleaves is what decides whether A beta is formed or not. For example, if you see here, the first enzyme which is acting on APP is beta secretase. If it acts on beta secretase, if the beta secretase acts on this APP and cuts it here, we get two segments. The first half here, it will be released extracellularly, that is your soluble APP beta, soluble amyloid precursor protein beta. And here if you see, this is your beta CTF. Beta CTF is nothing but beta carboxy terminal fragment, and this is the membrane portion and this will be the intracellular portion. Okay, now following this break by the beta secretase, now if gamma secretase separates these two segments, we get what is called A beta subunit and AICD. AICD stands for APP intracellular domain, nothing but amyloid precursor protein intracellular domain. So this is the intracellular domain which will be cleaved by caspase into JCasp and C31. So this is the segment or a subunit which is only made, which is the membranous portion. So now, as you can see now, so APP, amyloid precursor protein, is made up of extracellular portion, membranous portion and the intracellular portion. So when only the membranous portion is left behind, A beta subunit, and that is what accumulates. Okay, so now when C31 JCasp forms along with A beta, all three subunits are actually neurotoxic, especially C31 in combination with A beta subunit. No, these two are very toxic to the neurons, which will lead to cell death in Alzheimer's disease. Normally, how this APP is split is, first alpha secretase will act on this subunit. So when the alpha is acting, what happens is the membranous portion of the APP protein is split into half. So you can see here, you get what is called soluble APP alpha, soluble amyloid precursor protein alpha subunit, because it is cleaved by alpha. Here what we are getting is SAPP beta, because it is cleaved by beta secretase enzyme initially. So you can see here the extracellular domain also has a part of membranous portion and it will be released outside the cell. And what is remaining inside the membrane and the cell is alpha CTF, nothing but alpha carboxy terminal fragments are left behind. So now the gamma will cleave this. What we get is P83 and AICD. AICD, similar to this, what happens, it is cleaved by caspase to get JCasp and C31. But here you don't have E beta. This P83 doesn't have much function, it will be immediately degraded. Okay, so this is how normally the APP protein is processed. If you can see here, alpha secretase followed by gamma secretase and caspase enzyme. But in Alzheimer's disease, what is happening is it is first cleaved by beta secretase, gamma secretase and caspase. And also they tell this A beta protein also will be hyperphosphorylated. Similar to your neurofibrillary tangles tau protein which you find in Alzheimer's disease, A beta is found in senile plaques, right? So this A beta is also hyperphosphorylated, which will increase the neurotoxicity of this A beta protein. So this A beta protein forms the core of a senile plaque, which is a characteristic feature you find in Alzheimer's disease.